Dear friends, good morning. Wish you all Merry Christmas and Happy New Year. It is my proud privilege to welcome you on behalf of Professor Brahm Singh Horticulture Foundation, a not-for-profit organization, and my own behalf to this 5S webinar series, talk number 22. BSHF is thankful to NEPA, New India Publishing Agency, New Delhi, for sponsoring this webinar series. I'm happy to welcome co-organizers Dr. Preetam Kalia, ICR Rafi Ahmed Kidwai Awardee, former head Division of Vegetable Science and coordinator School of Horticultural Sciences, ICR IRI New Delhi, who is the speaker today. And the another co-organizer, I welcome him, Dr. Sharon Rajan, former director, ICR Central Institute of Subtropical Horticulture, Lucknow. As you know, today the webinar is on future prospects of nutraceuticals or nutraceuticals, whatever way we pronounce, breeding and fortification in vegetable crops by Dr. Preetam Kalia, the co-organizers of this series of webinar. Friends, the era of breeding vegetable crops for tonnage is receding, and biofortification of vegetable through breeding or crop improvement is picking momentum to be an important breeding program with the aid of modern biotechnology, AI technology, and others. We all know nutrients deficiency, disorders, and diseases in population throughout the world, where India figure prominently, are rampant and forcing us to consume chemical pills with every meal or in between meals. This may be a fashion also, but not a healthy thing. Pharma industry is entering the food industry. What a change. Our diet, our food should meet our body requirement, except in exceptional cases where supplementation is necessitated. Towards above goal, vegetable can play a major role by having vegetables or their particular varieties or hybrids, etc., to meet body requirement. Dr. Kalia would enrich our knowledge on this important subject with his very rich experience. I request Dr. Shailendra Rajan to introduce customarily. Dr. Kalia doesn't read any introduction. Who is known to this audience? As usual, question may be raised in chat box, which we will try to answer. Dr. Shailendra Rajan, sir.
Thank you very much. Good morning, ladies and gentlemen. It is a great pleasure for me to introduce our today's speaker, Dr. Preetam Kalia, a renowned vegetable breeder who is going to talk about future prospects of nutraceutical breeding and biofortification in vegetable crops. Our speaker has spent about almost his entire career of 37 years in developing dozens of outstanding vegetable varieties. He obtained BSc Agriculture, MSc Vegetable Science degrees from HPU Shimla and PhD in Vegetable Sciences from Himachal Pradesh Krishi Vishwavidyalaya, Palampur, Himachal Pradesh. He did his postdoctorate on Commonwealth Scholarship of the Great Britain from then National Vegetable Research Station, later known as the Horticulture Research International. So Dr. Kalia started his career from HPKV Palampur, where he rose to the level of professor before joining as principal scientist at ICAR IRI New Delhi. He took over as head of Vegetable Science Division, ICR IRI New Delhi and served for more than 6 years. He has served as ICR at ICR New Delhi. During his 3 years working in this web on a very important futuristic topic, advances in biotic research, resistance breeding, and biofortification in Indian cauliflower, nutraceutical mapping in carrots. He owns a long list of prestigious awards like the Kirti Singh Gold Medal Award, Himachal Shri, and Himachal Gaurav Awards. For his outstanding contribution to the vegetable sciences, he has he was awarded Vashvik Award, IAHS Shiv Shakti Lifetime Achievement Award, Rafiq Ahmed Kidwai Award of ICR. During his career, spanning over 3.5 decades, Dr. Kalia contributed significantly in the area of vegetable crops improvement, having 33 varieties of different vegetable crops to his credit. Registered genetic stocks, 2 of CMS cauliflower, 4 of CMS tropical carrots, one resistant to each of black rot and downy mildew diseases of cauliflower with NVPGR. His major contributions are in exotic vegetables, nutraceutical and resistance breeding and genetic mechanism facilitating hybrid breeding in cauliflower and carrots. Dr. Kalia is credited with introduction of new vegetable crops in India such as broccoli, leek, and also have honor of developing their indigenous varieties and popularize them. Dr. Kalia contributed significantly in disease resistance breeding, developing powdery mildew resistant golden pea variety Palam Priya, black rot, and downy mildew resistant cauliflower varieties Pusha, and Pusa Shukti. He established genetic mechanism cytoplasmic male stability in tropical carrot and Indian cauliflower, paving the way for the hybrid development. He identified genes which impart resistance to black rot disease in cauliflower, which are being bred into commercial varieties through marker-resistant breeding. Dr. Kalia has progressed beta-carotene enhancing OR genes in Indian cauliflower, which will go a long way in tackling beta-carotene malnutrition problem in India. Dr. Kalia has published more than 110 research papers in journals of repute. Dr. Kalia has guided 5 MSc and 6 PhD students. 3 of his PhD students were awarded Jawaharlal Nehru Award for Best Thesis. He is Fellow of National Academy of Agriculture Sciences, Indian Society of Vegetable Sciences, and Indian Academy of Horticulture Sciences. Dr. Kalia represented India in APO sponsored meet on quality and safety standards for fruit and vegetables in Taiwan. He also served as IAEA sponsored training expert on vegetable breeding to scientific staff in Mauritius. He has been elected as vice chairman of the International Society of Horticulture Sciences for the Division of Horticulture for Development from August 2018. Dr. Kalia is consulting editor for the International Journal of Vegetable Sciences, published by Taylor and Francis, USA, and served as chief editor of Indian Journal of Horticulture, published by Indian Academy of Horticulture Sciences for 3 years. Dr. Kalia visited United States, Kingdom, Japan, Belgium, Netherlands, Portugal, Taiwan, Australia, Mauritius, and New Zealand. In the connection with postdoctorate studies, he visited all these countries, presenting research papers in congresses, symposiums, international collaborative research project meetings, and for imparting training to the scientists. Ladies and gentlemen, please join me in welcoming Dr. Kalia as a speaker for this very, very important talk.
Okay. Thank you, sir. Good morning, everybody. Thank you, Dr. Rajan, for the introduction. Most respected Padma Shri Professor Brahm Singh ji, founder of BSHF. It's indeed a pleasure for me to speak on this subject, uh, future prospects of nutraceutical breeding and biofortification in vegetable crops. These are futuristic aspects, as already has been highlighted by Professor Brahm Singh ji. The improvement target in vegetable crops, especially, is now focusing on quality aspects rather than on yield. And therefore, for the last over 37 years, I had been focusing on these aspects, including the conventional breeding initially, and then following the marker assisted breeding to hasten the program. And today we uh, say to people to use the modern breeding tools, especially the genome editing tools, to further hasten the process of development of quality rich vegetables. And with that, I go ahead with the presentation. This is the first and foremost aspect to be told to the viewers that the horticulture production, the latest data which has appeared in July 22, says that the horticulture production has reached to 300, almost 342 million tons, and in that vegetable production has reached to 204 almost 205 million tons. So that indicates that the production has increased quite a lot. And as per the uh, ICMR recommendations for our dietitians, recommendations are that earlier 300 gram of vegetables must be consumed daily, comprising 100 gram each of uh, roots, leafy, and other vegetables. But now the level has been raised to 350 gram by the ICMR. That minimum, this is the minimum 350 gram, but upper limit, there is no upper limit, as we can go ahead with the to further get enriched with various nutrients that we have in these crops. As we say that the vegetables are naturally biofortified crops, they are the nutrients, they have the nutrients in that sector. So from the last year's 2021, the production was 200 million tons, and now it is predicted to be 205 million tons. So there is increase in production, which says that many small and marginal farmers are shifting to the cultivation of these crops. And we also are aware of that the consumption pattern, as because of the awareness about the nutrition of these crops, has also increased. Now the production level has increased tremendously, and therefore, in order to benefit the farmers with the increased production, the future varieties must, as indicated, that they must be rich in some other bioactive compounds. Especially we know that some secondary metabolites in plants are functionally active in human body, which we call them as nutraceuticals. And vegetables are, we know that they are good source of functional elements, and they act as protective foods. And because of the importance of vegetables in food based approaches, the potential therefore is to be explored as source of nutraceuticals. And the biofortification already has been done in many crops, and there has been success with respect to sweet potato, tomato, cassava, cauliflower, yam, because in certain locations where there is a deficiency of various minerals and micronutrients, and the populations are deficient, there uh, the adverse effects on the human being are quite a lot. Uh, the quality aspects, which includes nutraceuticals, are getting attention nowadays because of the lot of awareness that is being created, and most of the breeding programs are including these aspects for improvement. And most of the quality traits, as we know, they are complex in nature, in inheritance. Therefore, The molecular aspects as well as diagnostic tools are required for tailoring desirable genotypes. And also, with the recent developments in this sector, modern breeding tools, genome editing tools are becoming more desirable, and the shift is towards these. And as indicated, the vegetable quality improvement is a global requirement because of current evolution of consumers' demands, as I indicated. And the producing vegetables with enhanced nutritional and organoleptic quality is one of the most challenging targets for breeding, facing with climatic changes and the needs for more efficient production system. High throughput techniques, especially metabolomics, transcriptomics, and genomic advances, represent useful tools to identify genetic architecture and biochemical pathways, and also to predict breeding values for selection and deployment. In the past, mutagenesis and tissue culture has been employed for inducing novel variation, overcome various crossing barriers, and speed up the achievements of true breeding lines. And from 90 onward, marker assisted selection has hastened the process of selection. And the novel omics tools and the genome editing tools, as I indicated before, which are the recent one, are being used as cutting edge techniques to face old and new challenges in vegetable crops, with potential to increase qualitative value of crop cultivation and solve the problem of malnutrition over the next 30 years. Modern breeding tools to address global challenges, including climate change, increasing yield and quality to secure a global food supply for a growing world population, and participate in maximizing the resources of farming systems. Current tools, which we normally call as multiomics approaches, are a fundamental support for breeding to match crop metabolism genetics and further improve organoleptic and nutraceutical quality in new varieties. So that indicates that the new tools are to be employed, as indicated, that polygenic inheritance mostly for the organoleptic and most of the quality traits. So it's a cumbersome process, and we have to shift to the modern tools to hasten the process of development. One of the nutrient called as GABA, G-aminobutyric acid, which is present almost in all crops in various levels. Pumpkin has been found to have highest one. But the food and pharmaceutical industry in recent years showed a lot of interest for this bioactive compounds, which has been found to be of great potential for human health and consumer demand for health promoting functional compounds, which is resulting in a release of a lot of GABA enriched products. And therefore, because the tomato is one such crop which is most produced and consumed vegetable worldwide, and fruit bearing model crop has received much consideration for its accumulation of this GABA bioactive compounds. Although many different strategies have been implemented, from classical breeding to the induced mutagenesis, new breeding tools have achieved the best GABA accumulation results in red ripe tomato fruits, along with shedding light on GABA metabolism and gene functions. So that's very important, because the biochemical synthesis of most of the bioactive compounds that we have in nutraceutical kitty, and also the ones which are immediately required to be increased in new varieties, the help of the new techniques is tremendous, which helps in knowing the gene function, which can hasten the process of development of new varieties. So the nutraceutical, as I always talk about, that the term was coined by Dr. Stephen DeFlies in 1989, who was founder and chairman of Innovation Medicine Foundation. And nutraceutical is a composition of nutrition and pharmaceutical value. That means that nutraceutical is a food or part of food which provides medical or health benefits, including prevention and treatment of diseases. And the food grade nutraceuticals, the phytochemicals in general we talk about in fruits and vegetables, have phyto pharmaceutical value. They are naturally antioxidant, natural food colorants, and nutraceuticals and dietary supplements from protein, carbohydrates, vitamins, minerals, uh, amino acids, medicinal herbs, minerals, etc. And the functional foods which can be had having these properties, fortified foods, energy boosting drinks, sport bars, beverages, etc. So the natural antioxidant properties of vegetable crops helps in preventing or slowing down oxidative damage to our body and acting as a free radical scavenger. The classification of nutraceutical bioactive compounds, because it's very important for, as I say, that this is for the future breeders, the new breeders who are opting for breeding of vegetable crops. They need not to concentrate on yield enhancement. It is already there. We have varieties available, but how to enrich those varieties further with these compounds so that the utility of these crops not stays with the fresh consumption point of view, because fresh with the enhanced nutraceutical and bioactive compounds, the consumers will be benefited from fresh consumption also, but the extra produce which is available with the growers, that can be utilized by the industry, pharmaceutical industry especially for extraction of those compounds and making them available in different other forms. So the classification is into five major groups: carotenoids, phenolics, alkaloids, nitrogen compounds, organosulfur compounds. And the carotenoids, phenolics are major compounds, and organosulfur compounds which are available in some of the vegetable groups. The nutraceutical industry today, from the global market point of view, it stood in 2019 at 247 billion US dollars, and is expected to increase to next year to 336 billion dollars, whereas by 27 it is expected to touch 723 billion dollars at a compound annual growth rate of 8%. Whereas from Indian point of view, the in 2019 it was at 5 billion US dollars, and is expected to increase next year at 21% compound annual growth rate to 11 billion dollars, and with market share of 3.5%, and business segments in this functional food, beverages, dietary supplements, etc., etc. But what is the cause of rapid emergence or shift of people towards nutraceuticals and edible colors? Because mainly that people are dissatisfied with the drugs cost and conventional health care, which is not dependable, and also the synthetic colors which are being used in food, which are carcinogenic, and the chronic diseases with the poor therapeutic alternatives, rising health and wellness awareness. Large population try to get rid of the effects of aging and want to stay healthy and uh, not to age faster. And a new focus on preventive medicines is there. Desire for personalized medicines, self medication of most of the people, and public perception that natural is good. Therefore, the shift towards the for management of health through diet. So diet diversification is, of course, uh, going towards that direction. And the bioactive compounds, although they are present in very little quantity in crops, but the effect produced due to their deficiency on humans are quite large. And therefore, the major phytochemical groups which are of concern are carotenoids, which comprises of lycopene, lutein, zeaxanthin, beta carotene, polyphenolics like flavonoids, flavones, anthocyanins, glucosinolates, sulforaphane, indole 3 carbinol, thiosulfides, aline, methane, ethene, dietary fiber, lignin, pectin, cellulose, and miscellaneous other categories are selenium, phytosterol, and stanol, saponins, etc. So the nutraceuticals and their benefits, as for the crops, which are the vegetables. On the left side, we have vegetables which are rich in some of the phytochemicals which are indicated in front of each of these vegetables, and the benefits, especially from major problems that the society is facing from cancer point of view, and cholesterol variation, antioxidant property, damaging oxidative damage to our various tissues, diabetes and hypertension, and also cardiovascular problems. So that sort of thing are taken care of with most of the phytochemicals, the major problems that society is facing today. And the source of nutraceuticals and health compounds. On the left side, we have groups of vegetables: alliums, crucifers, solanaceae umbelliferae, compositae, legumes, cucurbitaceous, green leafy vegetables. And then the second column, what nutraceutical compounds or bioactive compounds these group of vegetables are predominant in, and the what are the health benefits that we can derive from them if we try to diversify our diet with these vegetables, and try to include in most of the diets that we have had morning to evening, to get rid of various problems, the day-to-day problems that we face with because of monotonous diet. And then here are the some of the benefits of these nutraceutical compounds which are present in plant system, what they do in plant and what they do in human health. So we find that they also have plant defense system. They play a role in defense system in plants, signaling molecules, the flavor and the pigments for attraction of pollinators. They contribute towards quality aspects because of various uh, actions in flavor, aroma, and then, as indicated before, and anti cardiovascular diseases. They act as anti-cancer problem, various cancers, eye health also. Like that, they have all the benefits that we can derive as indicated in the last column of this slide. And then specific vegetables with the specific active principles of different compounds is indicated here with the health benefit that we can derive from these specific compounds. For instance, in onion we have quercetin, which helps in improving anemic skin disorders and stomach cancer. Garlic, allicin, alline, anti-parasitic, anti-cancer, antiviral, and antibacterial properties it has. Likewise, lycopene, which prevents cardiovascular diseases. Capsaicinoids, antioxidant, anti-inflammatory. Vitacinin, which is for hepatic disorders. Refernin, antimicrobial, antiviral, bronchitis, and hyperlipidemia. Like that, carrot acid, oligosaccharides, which are anti-cancer, artery protecting, immunomodulating, infection fighting, disease, cholesterol, etc. And mucilage in okra for blood sugar stabilizing, also anti-ulcer, anti-cholesterol, bind to bile acid. So that is specific with some compounds in specific vegetables. From colors point of view, that we have red, orange, blue, purple, yellow, cream, green colors, which are because of the pigments like lycopene, beta-cyanine, beta-carotene, anthocyanin, lutein, chlorophyll. And these are the vegetables where they are predominant in, and which can be used for extraction of these, and using these uh, colors in place of synthetic ones. And there are some edible color-rich varieties which has been developed through conventional breeding program in the past, which are being used by some of the industries for extraction of these compounds. But we need to hasten the program to develop the future variety with further other characters also. In case of coal crops, the glucosinolates are the important nutraceutical bioactive compounds, which are organic compounds which contain sulfur and nitrogen and are derived from glucose and amino acids. And these glucosinolates convert to isothiocyanates, which is sulfur containing, and indoles, which do not contain sulfur. When vegetables containing them are cut, the glucosinolates convert into these major compounds, isothiocyanates and indoles. And the cruciferous vegetables, especially cauliflower, cabbage, broccoli, Brussels sprouts, are rich in these compounds, and they act as against cancer by phase 2 enzyme induction. And broccoli and cabbage show greatest protection against bladder cancer. Uh, their hydrolytic and metabolic products act as chemoprotective agents against chemically induced carcinogens. The types of glucosinolates that mainly we have: isothiocyanate, sulforaphane, glucobrasicin, gluconestrin, indole 3 carbinol, and sinigrin. And where they are predominant in different cruciferous vegetables is indicated here. And also that the glucosinolates are having two major groups: aliphatics and phenyl indoles. And in aliphatics there are 9 glucosinolate forms, and in phenyl indole there are 5. And while all these major coal crops were assessed for it, the broccoli was found to be predominant in glucoraphanin, and sinigrin and gluconapin was predominant in Brussels sprout, and in cabbage, cauliflower, kale, sinigrin was found to be predominant. So, knowing that, the breeding strategy to create new variability for quality traits, two major aspects: that production of genetic variability through conventional or non-conventional techniques, and the efficient selection within the variability created that has to be done. And in that sector, selection procedures need to be followed, which reduce time and cost are essential, especially when the breeding programs are based on interspecific hybridization, especially. Secondly, marker-assisted selection can be ascribed not only to use of markers tightly linked to target genes, which we call as a positive assisted selection, but also we can make use of these markers specific when wild donors are being employed in hybridization programs, so that negative selection to the traits which are unwanted can be carried out. The challenges and opportunities in health compounds are from the point of view that the consumption of vegetable crops for preventing many chronic diseases is documented and proved. Therefore, the necessity of developing new varieties with high bioactive compounds is a challenge and opportunity to the vegetable breeders. And the new crop plants with the specific compounds can be bred using conventional breeding or genetic modification techniques. More variation in the functional compounds found among cultivars or in plants within cultivars is good opportunity to breed new varieties with uniformly high levels of those compounds. And consumption of such compounds, such plants rich in these compounds provides more health benefits by creating them by eating them on a regular basis, and would also lead to quality of life and reduction of health cost of nation. So, while breeding vegetables for optimum levels of phytochemicals, already the efforts has been made in screening and genetic variation and developing novel varieties, especially in carrot, melons for beta carotene, anthocyanins, and onions and peppers higher in flavonoids, and utilization of molecular biological techniques uh, is to develop map and clone genes involved in regulation of carotenoids, ascorbic acid, and flavonoid synthesis pathways is tremendous and helping in knowing various gene functions. And the goal to combine improved human health benefits from elevated phytochemical concentration with food flavor, yield, and stress tolerance. So, the combining of all these in the future varieties is important to have better utilization of the crops. Uh, how we can make use of genetic variation, where we can make use of that, I used to talk many times, and which is the basic to make improvement, that primary gene pools, secondary, tertiary, and quaternary gene pools are important gene pools to drive genes of interest, while we make improvement depending upon where the gene of interest is available. And the common breeding programs, where the genes are linked with some markers, and the breeding programs are the conventional, the backcrossing process as indicated in the first category, and then gene pyramiding, pedigree breeding, as you see that the coming out with individuals which have combination of most the desirable traits as indicated down here with the stars. These are the individuals which have combination of the desirable alleles. And then recurrent selection, where different individuals with different compounds are entered into a hybridization program, intermating, and then coming out with the population where these individuals with asterisks down are the ones which have desirable combination of the alleles which are important for certain traits. So the overall tools from modern plant breeding point of view, that where we have uh, product opportunity, development of product from trait specific in the crop, and the objective for maximizing breeding program and native traits, and where we have objective of introducing or enhancing attributes. uh, we need to have a genetic knowledge of the crop, and then through the maximization of a breeding program, we go for genetic variation, phenotypic measurement, genetic associations, pedigree analysis, and then germplasm development, molecular breeding, double haploids, ultimately leading towards varietal development. And from the other category, where we introduce or enhance attributes through gene discovery, positional cloning, gene shuffling, and then through transgenic plant development, plant tissue culture, gene expression, also leads to development of new varieties with the desired traits. So these are the techniques, and the new breeding techniques, normally cisgenesis, intergenesis, and ODM, which is oligonucleotide directed mutagenesis, and agrofiltration, and others are like RDDM, which is RNA induced DNA methylation, reverse breeding, and the recent targeted mutagenesis mediated genome editing techniques, zinc finger nuclease, TALEN, and CRISPR-Cas9, where the ZFN and TALEN also overtook by CRISPR-Cas9, and CRISPR-Cas9 is still evolving one, where Cas9 protein is now being replaced by CPF1. So here in this slide, it is just the schematic illustration of ZFN structure and the principle of ZFN mediated genomic modification. And the target site of ZFN is recognized by the left and right half monomer, that each consists of tandem array of ZF engineered ZFPs, and each ZFP has the property of recognizing nucleotide uh, triplets, as you find here. And the ZFN monomer has N-terminal domain and C terminal functional domain. And when the recognition of the target site, that the target site is here, and the recognition of this target site, target sequence by the left and right ZFPs results in dimerization of the fork1 endonuclease, which is at C1, at C functional domain. And the DNA cleavage takes place here, where we have 6 base pair long sequence, and the induced DNA stand break here is repaired by the non-homologous end joining and homology directed repair, resulting into this gene mutation around the cleavage site, including nucleotide addition, deletion, and substitution. And similarly, in case of TALEN also, where the target side is uh, recognized by the left and right half monomer, each of consist tandem repeat of tail repeat, and each tail is further comprised of 34 amino acid unit, which differs at two hypervariable at 12 and 13 site, which is known as RVD, residue variable diuse, which determine the recognition specificity of each repeat. And the TALEN monomer is consisted of N-terminal domain and C functional domain. And the when two talents come together, the C1 C terminal functional domain has F1 fork1 endonuclease, and due to simultaneous binding of these two tails left and right, it results into dimerization of fork1 endonuclease, resulting into cleavage of the target DNA. DNA stand breaks takes place, which is repaired by NHEJ, and HDR, which are non-homologous end joining and homology directed repair, resulting into gene mutation at target site, including gene nucleotide addition, deletion, or substitution around the cleavage site. Then, with respect to this schematic illustration of CRISPR-Cas9 system structure and principle of CRISPR-Cas9 mediated genomic modification, here the synthetic guide RNA contains a region which has a 20 base pair length complementary target site on genomic loci, and it stems the loop synthetic guide RNA that mediates the binding of Cas9 protein, and the protospacer adjacent motif, which is here represented by red color, and the Cas9 protein is represented by the brown circle. The cleavage site here is located 3 base pair away from the protospacer adjacent motif, which results into DNA strand breaks of the target DNA, which is repaired by this non-homologous end joining and or homology directed repair, resulting into mutation at target site, including nucleotide insertion, deletion, and substitution at the target around the target site. So the first product using this technology was in cabbage, where the Swedish scientist called Jensen removed small part of cabbage DNA with CRISPR, and that codes for a protein called PSBS, which was called as safety valve in photosynthesis. And this was the first time that a edited plant was cultivated outside laboratory. And this is how they have indicated that the gene edited cabbage product was eaten. Then transfer of flesh color from the wild relatives for enhancing the lycopene content and the color, using parent as Solanum hirsutum, and the processing cultivar E6027, which has this red one as the nil, which is carrying QTL from Solanum hirsutum for red pigment. And similarly, in case of Chinese cabbage, the recessive OR gene imparts uh, enhancing the beta carotene content. And as you find that this is a wild parent, and this is the one which has been introgressed with the OR gene enhancing beta carotene content. Similarly, the PR gene mutation enhancing anthocyanin in cauliflower, which is a semi-dominant gene, and you find yourself where the gene has been introgressed, it has turned the plants purple, and all stages, including seeds, showing the expression of anthocyanins with the PR gene. And then nutritious tomato lines by AVRDC created long bag golden tomatoes, which are not yet popular. From that point of view, only people prefer red tomatoes. It contains 3 to 6 times more vitamin A than standard types, and just single improved tomato can meet out daily requirement of vitamins beta carotene. Similarly, the allicin, which is very important sulfur compound in alliums, through transgenic approach, also these allicin content has been enhanced. And then the approaches in breeding for quality attributes in vegetables. Uh, as we know that germplasm assessment and pre-breeding program to transport traits from different genetic backgrounds, and the search for promoters for increased bioavailability and reduced anti-nutrients in biofortified varieties, transgressive segregation or heterosis for quality traits, and molecular breeding for handling complex quality traits, transgenic approach for genes from alien sources, exploring gene editing approaches, as I have highlighted already, use of innovative diagnostic tools for handling micronutrient traits, and then consideration for breeding nutraceutical rich varieties. That the source germplasm must be high density of target nutrient. Biofortified variety must be having wider adaptability with high content of target nutrients. Processed forms of variety hybrid must retain sufficient quality attributes of nutrients, antioxidant, etc. And bioavailability of nutrients in human body must be high. Acceptance of biofortified crops by target groups, like farmers and consumers, are the two major groups that must be there. And then breeding approaches for biofortification. The conventional approaches has to be there all the time, keeping all those factors mentioned in previous slides in mind of germplasm. And then advanced approaches to hasten the process of integration of the traits of interest, knowing gene function, etc., would be important. And biosynthetic process is important to make use of these new tools. This is important to mention here that most of the institutions that we have in the country, especially IARI, IHR, IIVR, are now making use of all these technologies and ushering into the new utilization of the new techniques. But in the university sectors, the process is very slow. There are a lot of universities in the country, and until they also adapt these programs and come out with improved varieties at faster rate, it can be possible only if they make use of the new techniques, get their scientists having training in these areas, and proceed further for helping farmers and growers. Then approaches to target micronutrient malnutrition. There are different approaches. For short term, we make use of food supplements, uh, food fortification, which is a medium term approach. But the long term approach is biofortification, that I have highlighted, to get varieties which are rich in various micronutrients, so that the seeds of biofortified crops can be made available even in the far-flung areas where it's impossible to carry, or where it's very difficult to carry the fortified food items to provide to the population. And also that dietary diversification is also a long-term program that the people are made aware of or educated about the inclusion of different types of crops to get benefits of different nutrients in different background. So the biofortification for improved health, uh, especially in staple food, has been carried out through conventional agronomic management, mass genomics, and or genetic engineering world over, especially in the problematic areas, especially in Africa, Asia, Latin America, where there is a problem of micronutrient deficiency, especially the minerals, iron, zinc, and provitamin A carotenoid. especially in the children and women. Uh, these problems are very many in these Africa, Asia, and Latin America, especially. And due to the low concentration of these micronutrients in the foods consumed, therefore, biofortification of food crops is important, and it is being suggested to alleviate the deficiency of these important micronutrients in the society, to get rid of them from malnutrition. And human studies provide the most reliable indication of viability of micronutrients in biofortified crops compared to that of the animal and in vitro studies, which are from the point of view of absorption factors and screening varieties developed through breeding programs. And then results of human viability studies from the minerals point of view in biofortified and non-biofortified crops are consistent, but compared, I mean, in contrast, the bioconversion of beta carotene to retinol in provitamin A biofortified crops is from 2.1 is to 1 to 6.5 is to 1, was found to be more efficient compared to that of average conversion of 12 is to 1 in non-biofortified foods. Then uh, about the viability of micronutrients from crops are much different in case of minerals compared to that of carotenoids, as provitamin A is most affected by dietary habits and food processing. That by adding fat to the meal, it improves absorption, whereas food processing, especially the heating, results in substantial loss of carotenoid pigments, I mean carotenoids. And the bioavailability of the minerals, especially iron and zinc, is influenced mainly by the presence of anti-nutrients such as phytates, which can be reduced through breeding, but the balance has to be maintained so that it's there also, so that one can derive benefits which phytates provide to plants and humans. And to minimize the negative effects of food processing and other factors that adversely influence bioavailability, biofortification is important to increase micronutrient concentration in staple crops, and it continues to be the primary goal. So internationally, the efforts has been made in improving the I mean, the staple crops, especially maize, rice, cassava, beans, wheat, sweet potatoes, and pearl millet, where there are the major crops in the endemic areas. And the biofortified foods with relatively high micronutrient density have higher total absorption rates than the non-biofortified varieties. Therefore, the evidence supports that the focus on efforts to breed plants with increased micronutrient concentration in order to decrease the influence of inhibitors and to offset losses from the processing, it's important to have biofortification done. And keeping that factor in mind, India has in 2020 released 17 biofortified crop varieties across groups of crops, especially rice, wheat, maize, and finger millet, little millet, mustard, groundnut, yam in vegetable sector. So we find that through various studies, biofortification has resulted into the more absorption in the carrot, especially where the transgen CXS1 introduced, was found that uh, on consumption of 100 gram of fresh carrots, 45.9% higher for female and 38.7 higher for males uh, from those carrots expressing uh, SCX1 gene. Therefore, it's important that the bioavailability in calcium enhanced carrots was more compared to those of traditional ones. Similarly, for lycopene and beta carotene studies also indicated, as you find from the first graph, that where red carrots and white carrots were used, and from the baseline, the red carrot has bioavailability of lycopene tremendously, which is significantly better than the one which doesn't have. And similarly for beta carotene, as you find here, where it has been compared, that consumption of red carrots, white carrots, and tomato paste. So the red carrot availability of lycopene was significantly better even compared to that of tomato paste, which is also having uh, lycopene. And in the third graph, as you find here, where also the red carrots were compared with the white carrot plus tomato paste and red carrot has been found to be significantly better in availability of beta carotene compared to that of the other two categories. So that indicates that bioavailable crops are better for improving the health, as also yellow cassava has been studied from beta carotene point of view, as you find here that this is control and this is the supplement. So the availability of beta carotene from yellow cassava was much better compared to that of even supplements. And also the prevalence of vitamin A deficiency was less in those who were fed on yellow cassava. So the studies on different crops was carried out by us at IARI to for various nutrients point of view prior to initiation of breeding program. And as you find here from the data, a lot of variability, which is the basis for making improvement, has been obtained in all these categories of nutrients mentioned here in across five crops that has been assessed. And similarly, from phenolics point of view, wherever these phenolics were found, noticed, they showed variation, which could be useful for improving those traits. And similarly, for antioxidant properties through different methods, DPPH and FRAP and SOSA, also you find that the variability was found. There's a range of these nutrients that has been found, which shows improvement prospects. And similarly, from vitamins point of view, especially, of course, from all vitamin point of view, but from total carotenoids and beta carotene, which we mainly say, also others are important, of course, but we do find that there is a lot of variability, as you find in the crops and different varieties which employed for this study. So there is a win-win situation and positive situation and future uh, prospects for improvement of these. And similarly, fatty acids, which are very important for any crop growing and for nutrition, and as especially from linoleic and linolenic acid point of view. And you find that again there is a tremendous variability that has been noticed for making improvement, quality improvement crops in these. And the nutraceutical rich vegetable varieties developed by conventional breeding program. These are lot of varieties which has been developed, which are rich in various vitamins and other aspects. And the nutraceutical rich carrots were also developed as indicated here. So these programs were carried out especially by me while at IARI, and we came out with the specific compound micronutrient enhancement rich varieties, which are of course now you'll find as here. These are being grown by farmers, as Professor Brahm Singh told that we must highlight. So this is highlightation that these varieties Pusa in carrot, Pusa Asita, and Pusa Vristi, cultivated on commercial scale, and also the farmer-led cultivation of these crops being done by some industries for extraction of these compounds. and then understanding the genetics of the anthocyanins and the anthocyanins predominant, finding markers closely linked to the locus of anthocyanins and using it for marker selection. And similarly, the program for lycopene and beta carotene was also going on, developing different populations, and also the development of hybrids which are rich in these lycopene and beta carotene and anthocyanin. Similarly, in radish also the advancement was made at IRA itself, that we developed these Pusa Jamni, Pusa Gulabi varieties, and Pusa Hriday when I was Palm Hriday when I was at Palampur, which are also rich in ascorbic acid, especially, which is important for a salad vegetable, and anthocyanins and total carotenoids. And also identifying introducing the genes for enhancing beta carotene in cauliflower, where the native gene was employed, and using marker assisted breeding, we have been able to develop varieties in the third group. And the earlier group program is also in advanced stage, where beta carotene and lutein rich lines have been developed, and in the future we will have such material available for consumption by the consumers and growing by the uh, farmers. Biofortification of anthocyanins also in cauliflower was initiated at IARI, developing its inheritance pattern, where the dominant gene was found to controlling, and these are the parental hybrid and the segregating population, showing the different plants with a different level of anthocyanins. And these are the different segregating plants in the F2 generation, where the prediction of gene combination has been indicated. As you find here, both BOP1 and BOCAL, dominant homozygous genes, and here the heterozygous state of these genes, and then here in white both recessive homozygous category, and these are the other ones category where the gene prediction has been indicated for these different colors. And here the some primers have been screened, and this one of the primer was found polymorphic, this one BOMY B3. And this breeding program of this broccoli was initiated way back in 1987, after I returned as a postdoc fellow from UK. Initiated first the development of broccoli program, as I found that broccoli is important crop in UK, but we had not seen it before. So the first variety as Palam Samriddhi was developed from Palampur, and based on this variety we popularized the crop in the country. And today we find that the crop is available in every nook and corner. So the initial efforts in developing the first variety, Palam Samriddhi, and then popularizing the crop, telling farmers that this is important from nutrition point of view. So this is the early variety, Palam Samriddhi, and then for hills, the late maturing variety, Palam Hritika, we developed long back, and these were released. Palam Samriddhi was released in 95, and Haritika and the colored Palam Vichitra heading broccolis and Palam Kanchan were released in 2003. Some farmers in some locations for the colored ones are producing, but the green ones are now being grown throughout the country. And we also assess them from nutrition point of view, total carotenoid and ascorbic acid especially, and the other nutrients, these minerals especially, they were assessed. And the important factor recently we found with respect to Palam Samriddhi was that it was found to be salinity resistant. And in the Gujarat area, in the coastal areas, from the last 4 or 5 years, this is being cultivated in a big way, and where there was no crop used to be grown. As you see here, the Palam Samriddhi is growing luxuriantly, and farmers are getting benefited, and also the consumers are getting benefited from its nutrition. So biofortifying these crops further especially with the glucosinolates and this one of the species called wild species Brassica villosa was found to be higher in MIB 28, which indicates that the glucoraphanin and the biofortified beniforte broccoli shows a bioavailability of this glucoraphanin 27 70% compared to this that of normal broccoli, which has 100%. So and this is sulforaphane mechanism of tumor inhibition, that how it helps in tumor inhibiting. So that is the utility of this. And development of glucosalinity glucosinolate rich broccoli using Brassica villosa is the process indicated here. Then increase in glucosinolates content in broccoli using Brassica villosa, the same program. But here the use of linked DNA markers, as you find here that QTL1 and QTL2. And this is the picture of segregating populations where the this microsatellite markers uh, in QTL2 is indicated, which helps in identifying gene combination showing higher expression of glucoraphanin and glucoiberin. As you find here that in these three individuals, QTL1 and QTL2 were found in different genotypes, and the in QTL in the uniformly in case of QTL1, and there was variation in QTL2. Similarly, in heat-tolerant tomato nutraceutical content, especially lycopene, the variability was assessed 0.22 to 27.24 milligram per 100 gram in 93 lines, which were found to grow luxuriantly at Delhi in a temperature of May and June that we have there. Then beta-carotene content in tomato using wild relatives cheese money is indicated, where the near isogenic lines and F2 population were employed. And the improved varieties of sweet potato with orange flesh having higher beta carotene content and anthocyanin content, as we find Bhu Sona and Bhu Krishna, which are rich in beta carotene and anthocyanin both. And also the cassava variety Sri Visakam from CTCRI, rich in provitamin A to as high as 466 international unit per 100 gram. And the recently recommended biofortified varieties through in 2020 by Prime Minister's biofortified yam varieties Sri Nilima and DA 340 with enhanced zinc and iron and anthocyanin content, which were released by PM. And using the native gene from cauliflower for enhancing beta carotene of potato was also employed, which was found to be quite better compared to that of bacterial genes, which people are a little shy of using such material. Then transgenic carrots for novel ketocarotenoid production using beta carotene ketolase genes from Hematococcus pluvialis algae, where the new carotenoids astaxanthin, adenosanthin, adonirubin, canthaxanthin, beta-cryptoxanthin, and echinonone. Up to 70% of total carotenoid was converted to novel carotenoids with accumulation of up to 2400 microgram per gram root dry weight. And astaxanthin, adonirubin, and canthaxanthin were the most prevalent carotenoid in this. Then anthocyanin and flavonoids have also been investigated with transgenic techniques. And here, transgenic tomato line DL Rosa 1 expressing antirrhinum majus dalila Del and Rosia 1 Rose 1 transcription factor showed uh, purple color and an anthocyanin rich uh, phenotype uh, with respect to the peel, flesh, enhanced increase, and also the both AFT, ATV, and Rosia 1 Del transformed purple tomatoes had, besides improved in their nutraceutical value, extended shelf life, and lower susceptibility to post-harvest fungus botrytis scenario compared to normal tomatoes. So that is the additional benefit simultaneously, in addition to anthocyanin and flavonoid that were had from snapdragon genes into tomato. And that's here we get that with the further getting different anthocyanins like delphinidin 3, petunidine, and malvidin. These were predominant anthocyanins that were found. Then the recent genome editing tools provided novel functional insights in gene controlling, directly or indirectly, nutritional and organoleptic quality traits in various crops, and hinted, at least in some cases, at the possibility to readily translate new knowledges and genotypes in applied breeding. And based on the fact that tomato fruit color depends on accumulation of specific compound directly related to organoleptic flavor and aroma and nutritional quality. It was Lee in 2018 who selected 5 genes, SGR1 and LCYE, LCYB1, LCYB2, and BLC, involved in lycopene biosynthesis and its conversion to keratin. So this is how the those 5 group of genes, lycopene 1, 2, 3, 4, 5, where single gene, 2 gene, 3 gene, and 4 genes were involved. And we find that lycopene 1, where single gene had highest lycopene content, and then lycopene 4, where 3 genes were involved, is followed. And similarly, the beta carotene content in lycopene 1 and 3 and 6 was high, where we had different combination of genes. It was assessed by HPLC, as you find here, HPLC peaks for lycopene in the first category, and the breaker 7 tomatoes were used for analysis of these. So identification of different markers for quality improvement in vegetable crops, in different crops, as you find here, for different rates: high carotene, anthocyanin, glucosylation, pungency, minerals, carotenoid, anthocyanin, and these are the different genes which have the different markers. So it isn't the process of marker-assisted selection for the future breeder once you have the genes and the markers attached to it. So we can conclude from this that as the importance of regularly consuming vegetable crops is prevailing, it's important that we need to go for higher content of these bioactive compounds, and this is a challenge and opportunity for breeders. And also that the new crop plants with specific compounds can be bred through conventional plus the modified breeding tools. And more the variation for fractional compounds, which better is the opportunity for enhanced bioactive compound varietal development. And these plants will help the consumers for providing them better health and also reducing the cost of uh, uh, health of the nation. And the it will encourage the indigenous nutraceutical industry development and will pave the way for contract farming, benefiting farmers. As I indicated, for black carrot, the contract farming of our product that we developed is going on. And the anthocyanin concentrates are being extracted and exported by the Niger by Phytochem in Punjab. So biofortification is the long-term process. As indicated, it's important so that seed of biofortified crops can be made available in far-flung areas, and the vegetables for target nutraceuticals and develop holistic program through public-private partnership mode, and use involving breeder and health experts would be better. Focus is required on indigenous underutilized vegetable crops for their biochemical analysis, nutritional profiling, and utilization, and laboratory analysis for quality traits need to be strengthened. Introducing of nutrient-rich viable vegetable crops from the world over, new crops which are rich in different bioactive compounds can be introduced and brought to cultivation. Exploitation of wild species and land races for gene and QTL identification for nutraceutical, and emphasis is to be laid on use of molecular markers, molecular tools in conjunction with conventional breeding for developing vegetable varieties high in nutraceutical. Use of omics approaches to understand the complexities of nutraceutical biosynthetic pathways to tailor them as per requirement is important. And the genetically modified crops also overcome some of the nutritional deficiency, especially carrots. As also I gave one example of enhanced calcium level bioavailability is more, and it can take care of the osteoporosis. And fortified transgenic lattice with the zinc will overcome the deficiency of this micronutrient that severely impairs organ functions, and folate deficiency, which is also uh, coming becoming problem nowadays, is regarded as a global health problem and can also be overcome with the transgenic tomatoes with folate levels that provide a complete adult daily requirement. And transgenic lattice with improved tocopherol and resveratrol composition may prevent coronary disease and arteriosclerosis, and can contribute to cancer chemopreventive activity. So finally, the omics and genome editing prospects that we see today through domestication and plant breeding process over the years for enhancing yield and productivity, we have lost many quality traits due to genetic erosion and reduction in those traits which provide taste. And we see, listen from various people that in today's varieties and hybrids that we have in the market do not have that taste that used to be in the land races. So, but the new studies where the combination approach of genomics and metabolomics are showing the revert back that there is increase in diversity in aroma compounds in cultivars by making use of these modern approaches, so that we expect that in future we will retain those organoleptic taste again, and we will enjoy the benefits of the past land races. And advances in omics technology make it possible to elucidate genetic and molecular basis of the multiple traits composing quality using genetic, genomic, and metabolomic approaches, particularly in case of pleiotropy and unfavorable trait association. The application of CRISPR-Cas, the new form, not CRISPR-Cas9, but because various forms of CRISPR-Cas has evolved, and CPF1 in place of Cas9 has helped. CRISPR-Cas in general is helping tomato breeding improvement program because tomato is economic and widely cultivated crop, so that is the primary uh, crop, fruit crop as a model crop. And a similar technology could help in hastening or fastening breeding and reducing the gap with other model system crops, such as edible inflorescence in case of cold crops, can hasten the program of nutritional development in those crops. So with that, I thank you very much for the patient hearing. Of course, I have taken more time because of the different two aspects that we have had.
Uh, thank you very much, Dr. Kalia. I don't know how to say, say a few words about your talk. You know, we have got scriptures, Ramayana, Mahabharata, and whatnot. Not to make it too long, you have given us the Gita of biofortification of vegetable crops. वो बोलते हैं ना हिंदी में गीता का सार है, तो आपने पूरा सार दे दिया हमको न्यूट्री क्रिटिकल और ये पढ़ाने का, और कितना पोटेंशियल है, क्या है, कुछ छोड़ा नहीं है आपने। एंड वी आर होपफुल एंड वी आर प्राउड ऑफ person like you, breeders like you, doing the yeoman service to the country. Hmm? Great, great, great contribution. Anyway, I don't want to give another lecture because we are exceeded the time. Dr. Rajan, could you kindly briefly add to this? But will be damn difficult. I know that what to say, what not to say. But I think few points you can say.
So thank you. But it's a very, very important lecture given by Dr. Kalia. He has given several examples which are new to most of us. Might be are interested in those. I will only pinpoint flags those, as he has given example of GABA improvement in tomato through different techniques. Then he explained beautifully how the nutraceuticals are important and what are the different components. Then how the extraction of the bioactive compounds is important, global status of the nutraceutical industry, and how the vegetables can substitute it. Then edible colors, their importance, major bioactive compounds he has grouped. I can't give the details in a very short time, but most important was the breeding strategy for the development of the varieties. Genetic, its genetics, then markers and selection process, and the most of the compounds related with the as a anti-carcinogenic like glucosinolates, isothiocyanates, and many chemoprotective compounds. So he has given a great detail of the breeding strategy also, how the quantitative changes are required in phytochemicals in the vegetables. He has talked about it. Tools being used as a for a modern breeding, tools which are being used for enhancing phytochemical enriched components, and especially details of the new varieties he has given. He has given the schematic illustration of this Stalin, CRISPR, and TFN modifications. Then application of CRISPR in many of the crops he has given cabbage, lycopene, and how the solanum for increasing the pigmentation in tomato, PR gene mutation, anthocyanin, chloriflower, then golden tomato. Many examples are there. I can't list all of those. But he has also touched about the micronutrients in vegetables, how those are important, and effective bioconversion of the beta carotene into retinol. He has talked about it. Bioavailability is most important, particularly for the fortified variety. He has touched that also, and how the processing is important, and how we can have the varieties with a higher content, which can pass the processing. Then he compared different vegetables for all these components. Particularly, he talked about the colorful varieties, vegetables, varieties which are not particularly in general colorful, the colorful cauliflowers, carrots, cabbage, broccoli, rich in bioactive compounds. And he also talked about the transgenic development which is taking place with different technologies to improve the nutraceutical value. So we had a very beautiful talk with the colored vegetables are not only important, attractive, tasty, but also important for human health and beauty, particularly through their bioactive compounds. Thank you, sir.
Thank you, Dr. Rajan.
Thank you.
There are a lot of appreciation for his talk, but no question. Uh, People are not able to raise the question because he made the talk so clear and so vast, what to ask, what not to ask. A very vast topic, very important one. But one should remember few points what I like to highlight, that your vegetable may have the nutrient, good, but whether they are available to your body, important. We talked about that availability of nutrients to the body, very important point, and linked it with the nutraceuticals. Very important aspect here. Uh, my good God, what is that? The host has unmuted your mic.
नहीं नहीं
Are you
चल रहा है
able to
सर
hear
ठीक है.
me? ठीक चल
यस
रहा है.
सर यस सर.
ठीक है.
हाँ ठीक है.
Okay, okay, okay. Uh, so there is a vast possibility to improve upon and what I said in the beginning, that instead of having the pills in the diet, have these vegetables. What a vast range of things. Dr. Kalia has shown you the possibility, all sort of things. A huge see prospects of that. So why not to resort to that? I'm sure the planners will take note of it, and I'm happy that the academicians must have been very happy because his talk was totally academic with the practical implication reaching to the farmers, and people are getting benefited. I suggest here that the vegetables developed through these programs or would be developed in these programs, they should get priorities in that what you could say the midday meal or some other program, and particularly on the, because these days I am visiting hospital quite frequently. So on a hospital menu, there should be there the disease specific or the patient specific food or diet has to be prepared, because the vegetable or the food constituent or for nutraceutical will have better effect than the pills. What I feel. Last of scope and future, as he has himself emphasized, is for this. People won't go for that how much you are eating. People will ask what you are eating, which nutrient. He mentioned a number of acid, the omega fatty acids, which he has named, which are in great demand these days. And about the 50% of the population, ladies, uh, most of the ladies are deficient in iron. So Fe availability, if somebody can just solve that problem, nothing like that. Then zinc deficiency is making a comeback. Uh, a lot of scope, very important talk, Dr. Kalia. We value it, and you have narrated. You have worked very hard. We congratulate you for that, and we want such type of talks, and I'm sure the academicians must have been happy, and they will take advantage of this, whatever little service from BSHF and V3, and with the help of all speakers we are doing, hope it will be useful to the viewers. We will come back again on 31st, the last day of the 2022, with some beautiful topic. Whether the flyer is ready, madam? Yeah, this is the one. Hanging greenery and vertical garden, the beautiful one. We want to say goodbye to the 2022 with the beauty of horticulture, that is flowers, and not in an ordinary way, in a technical way, sophisticated way, and a futuristic way. Please do attend the last lecture on 31st at 11 a.m. With these words, I thank profusely to Dr. Kalia, Dr. Rajan, and Ms. Shivangi, and all others who have helped us in this conducting this program. Thank you so much. Meet again on 31st. Thank you.
Thank you, sir.
Thank you, sir.
Thank you, Shivangi.
Thank you, sir.
Hello.